Hi, good afternoon. Hi. Thank you, Aidan. Viviana, thank you very much. Thank you for this invitation. I'm very delighted to be here. And thanks to all these wonderful women that spoke and presented their talk. And I would like, you will see during my talk that I also work on female animals, female rats. It's not that only finding 50 great women, it was a hard mission. There are plenty of wonderful women, but even studying mechanisms of learning and memory in female rats, for example, is really missing. And we are focusing mainly on males, and I will present this data that we start to accumulate. So as Dan said, I'm going to talk about fear. Fear is very simple in terms of emotion, but it is very, very necessary for our survival. This is not the right presentation later on, but it's okay, I will manage. So if I will ask, where have you been on the 24th of October last year, I think no one will remember. Sami, do you remember where have you been on 24th of October last year? Okay, so however, if I will ask where have you been on September the 11th, when it happens, 2001, I think no one will ever say that I forgot where I have been, because this was a very intense emotional experience. So we all remember because there is one structure that is implicated, or an emotion that is developed, which is fear, which is a very vital response to physical and emotional danger, and is crucial for our survival. So always I ask my student not to think in terms of humans, but to think in terms of animals. And imagine that animals do not feel fear, so the chances for survival and to protect themselves, it's becoming low. So when fear becomes pathological, this is what we are studying in the lab. So in the lab, we study fear conditioning and extinction, and in order to better understand the mechanisms of fear conditioning or fear formation, we need animal models or preclinical studies. So in the lab, we use the fear conditioning paradigm, in which we associate or we pair between a conditioned stimulus, which is a neutral stimulus like a tone or context, together with electrical shock. So after pairing of these two stimulus for even one time, so there is a formation of long-term fear memory, which is expressed in an increased freezing to future presentation of this stimulus, the tone or the context. Now this fear memory can last for lifetime. Even in the life of animal, we can come and examine 30 days later on, and we can see that there is still a freezing to the presentation of the context or the tone, and this is the long-term fear memory. So as I said, fear memory is robust and long-lasting and can last for lifetime. Now the neural circuit which mediates fear memory is mainly composed of this tiny structure that is called the amygdala. And the amygdala is very important for the survival and for the execution of the emotional or physiological responses that are associated with fear, like freezing, like increase in blood pressure, increase in hormones, and it also modulates the emotional type of memories. As I said, fear responses can really last for lifetime. However, fear is not something that is very economical for the body, and it's not adaptive. So the animal needs to adapt when the situation does not predict any longer any danger. So we are now talking about extinction of fear conditioning, which relates to the idea that when we present after conditioning, when we present the animal again and again with this stimulus without the appearance of the electrical shock, then fear responses will gradually decrease, and thus we will have a decrease in freezing, and this is extinction of fear conditioning. Now, while fear association, as I said, can be formed even in one trial of association between context and electrical shock can cause fear memory that can last for months. Extinction memory, on the other hand, is formed after several trials, and it's not permanent because fear can always be relapsed. Now, when we are talking about post-traumatic stress disorder, then we are talking about symptoms that is characterized by pathological acquisition, expression, and persistence of learned fear association, or in other words, more fear and impairment in fear extinction. Now, the neural circuit, as I said, that mediates fear memory is the amygdala, which is very primitive and it did not evolve across evolution. And when we want to reduce the fear responses, then we are talking about the prefrontal cortex that was mentioned by Thelma. And there are close anatomical connections between the amygdala and the prefrontal cortex, suggesting that they interact and they can modulate each other. Now, the prefrontal cortex is very important, as we know, in executive functions, in empathy, in feeling the others, in social memory. However, it has a very significant role also in extinction of fear conditioning. Now, within the prefrontal cortex, there were detected subregions that they are very important for extinction memory. And here you can see that there is the infralimbic cortex, which encodes the memory of extinction of fear conditioning, meaning that if this structure is impaired, then we can extinguish, we can reduce our fear, or the animal can reduce the fear responses on extinction training. However, on the next day, we need to repeat this over and over. Now, how the infralimbic cortex or how the prefrontal cortex affects the amygdala in order to have extinction. So many hypotheses were reported. This was not supposed to be in my new presentation, but I have it here, so I have to explain it. So there are connections between either the infralimbic cortex inhibiting a structure that does not excite the central amygdala, which is the executive center of fear output, or alternatively, it activates GABAergic or inhibitory interneurons, and these inhibitory interneurons inhibit the center of fear responses. However, regardless of which of these hypotheses is the correct one and how the prefrontal cortex inhibit the amygdala, a very simple hypothesis could be delineated. That during fear-provoking situations, the amygdala, it's a very simplistic model, but during fear-provoking situations, the amygdala takes control in order to assure defensive behaviors and thus survival. However, as the situation changes and there is no danger, then the prefrontal cortex becomes dominant, and the emotional response that is elicited by the amygdala will be suppressed, inhibited, or in other words, extinguished. So the suppression of fear then will be impaired in cases in which the dominance will switch from the prefrontal cortex back to the amygdala, and this is indeed the post-traumatic stress disorder. And if I remember the first interview that I read about Sami, and from that I know him, that you were fascinated by these monks that they do meditation. You remember, Sami? They do meditation, and they found that these monks, they have more activation in face of emotional stimulus. They have more activation of the prefrontal cortex and less activation of the amygdala, and thus they are not anxious all the time. So if we talk about then fear and extinction, then we talk about dominance between the amygdala and the prefrontal cortex. When we are frightened, then the amygdala is more dominant, and when we are not frightened anymore, then the prefrontal cortex is dominant. Now, can we play with this dominance and make the prefrontal cortex more dominant or the amygdala more dominant? And of course, and as Thelma said, there are maybe Thelma mentioned transmagnetic stimulus, TDCS. So they can stimulate maybe the prefrontal cortex in order to promote extinction. However, this is, as I said, extinction is more hard to get than causing the animal being more anxious, and thus we we use this strategy of making the animals more anxious, and then we look at the extinction or prefrontal cortex dependent tasks. So what we have found is that increasing putting the animal on elevated platform, it's like bringing you and putting you, if you have phobia from heights, then you feel anxious, and we can see that it is mild stressor. It increases all the measures of physiological stress, blood hormones and adrenaline and corticosterone, and the animal is completely frightened. And then we can examine other behaviors, so we can examine how this exposure to stress affects behavior, electrophysiology, morphology, biochemistry, and molecular, and so on. And so we know that stress impairs or abolishes spatial memory, object recognition, these tasks that are related to the hippocampus, and also it abolishes or impairs extinction of fear conditioning, and thus because we are more occupied in survival and we cannot focus on cognitive tasks. We know also that exposure to stress is related to changes, morphological changes in the prefrontal cortex, and even one episode of stress is related to morphological changes. So exposure to fear or traumatic experience increases amygdala activity, decreases functions that are related to the hippocampus and prefrontal cortex, because the brain and body are busy to survive and not busy to acquire new information. Now, we know that several pharmacological tools attempted to see whether we can facilitate extinction and make us less frightened. However, we ask in the lab a very simple question. Can extinction be enhanced in a behavioral paradigm? And then Bal talked about empathy. And here I'm going to talk about how to treat fear by social support in rats. So we developed, in my lab, we developed a paradigm of social, it's called social extinction, in which we fear condition the animals as singles. And 24 hours later, we retrieve the fear memory, and we know that these animals are frightened. However, after this retrieval, we continue with extinction, either in a single condition, or we put animals in pairs. So they extinguish together. So these two animals went fear conditioning together, and now they are extinguishing together. And what we have found is that this extinction resulted in pairs, resulted in facilitation of extinction, meaning that they reduce their fears more easily than in the single condition. Now, the prefrontal cortex is very implicated in social behavior, and we know that oxytocin also, Viviana talked about oxytocin. We know that oxytocin is also involved in social memory. So the question that we ask whether this facilitation of extinction, this enhancement of extinction, can be related to prefrontal cortex oxytocin. And what we did, we blocked the oxytocin in the prefrontal cortex, and we saw the effects on this typical social behavior or reduction of fear. And we have found that when we do this in adult animals, male animals, we can see that it blocks the effects that is induced by being in pairs, suggesting that social facilitation or social support is mediated by prefrontal cortex oxytocin. Now, another thing of the prefrontal cortex is that it evolves during development, which means that if I can see that students here that they are below 26 of the age, so they have still ongoing maturation of the prefrontal cortex. And thus, studies have shown that the prefrontal cortex is similarly engaged in juvenile animals as compared to the adult animals. And for us, we thought that this is something that is not possible. And we have lots of data that shows that the recruitment of the prefrontal cortex is different in juvenile animals than in adult animals. However, in this experiment, what we have asked whether we can get similar effects of these social effects in juvenile animals. And we also asked whether juvenile or female rats will show similar effects. So as I said, studies on female animals is neglected so much, even though that animals, females, we know that women, especially women, they are more susceptible to anxiety disorders. However, all the studies are conducted in the male animal. So what we did was we blocked oxytocin in the juvenile female and in the juvenile male. And as you can see, while in the male there was no effect of this blockade, the treatment did not affect extinction. In the female animals, you can see that it blocked extinction and it blocked also the social support by extinction, which means that animals, when prefrontal cortex oxytocin is blocked, then the social support is not beneficial on extinction. So so this means that if you remember, Liron, where is Liron? She's not here. She's guilty. So if you remember, because I put in my new presentation also the figure of adult animals, you can see, if you remember the effect in adult animals, so also in the effect in the adult animals, you can see that it is similar to the juvenile female animals. So here we see that the juvenile male and the juvenile females are distinct, and that the juvenile female is more similar to the adult male. And this is something very revolutionary, because because I always say, if we needed a scientific experiment or scientific evidence that girls are more mature than boys, then I think I'm presenting here, and I will show you further evidence that female juveniles are different from male juveniles. So we went to social recognition memory, which doesn't include any aversive component, and it is very emotional for the animal, because the animal is exposed in the first training session to an object and to another animal. And normally, because rats are very social, so they explore the social animal more than the object. Okay, 24 hours later, we expose the animals to a familiar one and to a novel animal. And now the animal, if it has a good memory, then it will explore the novel one and will less explore the familiar one. So this is social recognition memory. And what we have found is that when we block the prefrontal cortex in the juvenile animal, you can see that we block social recognition memory, and they are no longer can discriminate between the familiar one and the novel one. Now, when we look at juvenile females, okay, juvenile females, you can see that in juvenile females, the same treatment does not affect social recognition memory. And also, when we test adult males, we can see also that it does not affect social recognition memory. So these results, again, using another paradigm, they show that the juvenile female has similar mechanisms as the adult male, and they are distinct from the juvenile male. So here I present all these data that they compare between these two paradigms, and you have the adults and you have the juveniles, females and juvenile males, and you can see that the juvenile females have similar mechanisms like the adult males, and they are distinct from the juvenile males. So to summarize, here we developed an experimental setting that shows that fear can be reduced by social support. We confirmed that the beneficial effect of social support on extinction is dependent on oxytocin. We also showed that these results, both with oxytocin and anisomycin, may suggest that even at prepubertal stage, before sexual maturation, Substantial differences exist between the juvenile male and juvenile females, and this should be taken into consideration when treating girls and boys suffering from anxiety or social disorders, because they have completely different mechanisms. So at the end, Nbal said that it is a group effort. So as you can see, it is really girls' power. And we have female-only women, wonderful women in my lab. And in order to have a boy in my lab, so we went to India, and we brought Kuldeep with us. So I thank my students, because without them, because I'm sitting in my office and I'm doing paperwork, and they are doing the real work, and this wonderful woman. And here I would like to say hi to Ilham Taha. Also, she finished PhD in our department, and now she is at ELSIC here. So wishing her very good luck in her career, and thank you very much. Thank you very much.